In this video, we will discuss about some of the FDA approved CAR-T product. So before going directly to this design of the FDA approved CAR-T product, let us quickly revise about the CAR-T cells and different generations of the CAR that we have discussed a couple of times in our previous videos. Let us just quickly revise that thing. So in order for the T cells to have the cytotoxic effect against the tumors, uh, it needs to have the information about the tumor's antigen, and that antigen information is usually provided by this MSC. So there is major histocompatibility complex that is present on the tumors will provide the antigen information to the T cell receptors that are present on the T cells, and along with that the other interactions will also occur, such as CD4 and CD8 also interact with the tumors cell surface, and that will lead to the T cells activations. The detailed process we have discussed in our old videos. You can refer to those videos to understand how what are the other signalings that also needs to be activated for this T cells activations. You can refer to the I button that can also take you to that video. And so this is how the T cells activations occurs uh, in human beings. So what researchers has done is they utilize this idea and made a CAR T product, such as the CAR means chimeric antigen receptors. That is, they transduce the receptors informations of the tumors into the T cells. So this, and at the same time they modify the intercellular domains of the T cells in a way, so in a way that whenever the cellular domains come in contact with its antigen, or then it will lead to the activations of the intercellular part, and which will cause the cytotoxicity proliferations of the T cells. So in the first generation T cells, they have only CD3 zeta that will lead to the proliferations, but it does not have that much efficiencies, thereby it does not give too high clinical results. In the second generations, they introduced a intrastimulatory domain, that is 4-1BB or CD28 along with the CD3 zeta, and in the third generation they use two co-intrastimulatory domains, such as 4-1BB and CD28 and CD3 zeta, and in the fourth generations they use a cytokine inducer also. So by default the T cells does not have the lytic granules release capacity. So in order to make T cells to release those lytic granules, uh, they have modified the intercellular domains of the CAR so that it can also release cytokine inducers. And in the fifth generations, the along with this intrastimulatory receptors and CD3 zeta, they have this interleukin 2 receptors beta. So it will trigger other signaling pathways such as STAT3, STAT5, and JAK, which will help in the proliferations and survivals of the T cells. And most of the FDA approved CAR-T product belongs to the second generations. So the product that we will discuss are the second generation's CAR-T product. The first one we will discuss in the which belongs to the category of B cell lymphoma or leukemia, or that targets these cancers. So in all four of them will target the anti-CD19, which is commonly expressed on the tumors, this B cell lymphoma and leukemia. And in the intercellular domains, one of them has these three CD28, two CD. We can say that in the hinge region also it belongs to the part of CD28. Transmembrane region is also CD28, and the co-stimulatory domain also CD28, and the T cells activation domain CD3 zeta. And for this another drug that is brexucabtagene autoluel cell, it will have the all the three CD28. It is similar to this the previous case, but I think it belongs to some different. There is some it might belongs to some different company. I'm not sure about the history of this, but from the figure it looks like both of them are same. In the third case, they use, instead of CD28, they use the hinge from CD8A domain, and the transmembrane region is also from CD8A. And in this case, they use 4,1 beta beta, co-stimulatory intercellular domain, and the CD3 zeta for T cell activations. And in the fourth case, they use the hinge from IgG4, and the transmembrane domain is CD28, and the 4,1BV, which is the co-stimulatory intercellular domain, and for activations they use CD3 zeta. So all of them are been approved by FDA for, and some of them are even considered for first line use, but based on their results, they are some of them are being used for like second line treatment. And in the case of multiple myeloma, what they do is they use the antigen binding domain as is anti-BCMA or dual anti-BCMA, and the other part, intercellular part or in the hinge part, remains same. That is CD8 belongs to the hinge part, and CD8A belongs to the transmembrane region. 4,1 beta beta is the co-stimulatory domain, and CD3 zeta is for the activations of the T cells. So this is some of the FDA approved product that are currently being used against the patients who are affected with B cell lymphoma, leukemia, and also CAR-Ts are being developed against this multiple myeloma. I hope you understood what are the differences in this CAR-T, and what is the history behind CAR-T formations. And researchers are been working on to make the other future generations uh, compatible for the direct use with the patients, that is getting it FDA approved. I hope you understood this CAR-T product, the brief history of CAR-Ts and the different FDA approved CAR-Ts. If you need to understand more about them in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.